Well, good afternoon, evening, everyone. I'm Rick Lifton. I'm the president of Rockefeller University, and I'm absolutely delighted to welcome all of you to this evening's presentation. I'm delighted to see so many of the university's benefactors and friends here with us tonight. The fall program is hosted by the Rockefeller University Council, the Committee on Trust and Estate Gift Plans, and the Women in Science Initiative. More information about these initiatives is provided in your program book. We have a wonderful program for you tonight, but before introducing our speaker, I want to acknowledge all of you whose generosity sustains the pioneering research initiatives here at Rockefeller. Your support enables us to recruit the most gifted and original investigators from around the world and provide them with the scientific freedom and resources they need to pursue their audacious research programs. Thank you so much. If this is your first visit to campus, I want to extend a special welcome and share with you a few of the remarkable things about this university. So Rockefeller was founded in 1901 by John D. Rockefeller with a singular mission, science for the benefit of humanity, and an unrelenting focus on excellence, both of which remain hallmarks of the institution today. John D. Rockefeller founded this institution recognizing that the surest route to developing effective ways to prevent and treat disease would be to understand their fundamental causes, and this prescient insight remains true today. An important element in the university's success as an engine of discovery is its unique and interdisciplinary structure. The university deliberately maintains a small size compared to other leading biomedical research institutions around the world. There are currently 72 heads of laboratory at Rockefeller with no departmental or disciplinary distinctions. We strive to recruit the most innovative talents from around the world and give them the resources, a great scientific environment, and time to develop their ambitious programs. And the results truly speak for themselves. Rockefeller scientists have made some of the most important discoveries in the history of biomedicine. These include the discovery by Oswald Avery here on campus in 1944 that DNA is the chemical of heredity. This discovery has been the key foundational discovery that has transformed the life sciences, the understanding of disease, and the development of new insights and therapies for diseases ranging from heart disease to cancer to infectious disease. So while we have only had 264 professors in our history, 26 of them have won Nobel Prizes in medicine or chemistry, a number not approached by any other institution anywhere in the world. And these include 6 Nobel Prizes in the last 25 years. And one of the elements that I think is striking is that these have been presented for work in diverse areas, including in cell biology, where Gunter Blobel was recognized for his discovery of how newly synthesized proteins are targeted to specific locations inside and outside of cells. For work in neuroscience by Paul Greengard, who showed how the activity of neurons in the brain is not simply regulated by electrical activity, but is modulated by hormones and other signaling molecules. For audacious structural biology work by Rod McKinnon, who came to Rockefeller and solved the first atomic structure of an ion channel. Ion channel activities regulate a wide range of cellular behavior in every cell and every form of life. In immunology, where Ralph Steinman discovered dendritic cells, which present pieces of proteins made by viruses and other pathogens to cells of the immune system, initiating the adaptive immune response to viruses and other pathogens. In genetics and neuroscience, where Mike Young used these tools to discover the molecular mechanisms of circadian rhythm, which synchronizes our sleep-wake cycle and metabolism to the 24-hour period of the Earth's rotation. And in infectious disease, where Charlie Rice discovered how the hepatitis C virus replicates, which led to the development of drugs that cure this often fatal disease, which affects 3 million people in the United States alone. And just this fall, we celebrated another milestone when Svetlana Moisov became the 26th Rockefeller scientist and the first woman to win a prestigious Lasker Research Award. Svetlana received the Lasker DeBakey Clinical Medical Research Award for her discovery of the hormone GLP-1 and the elucidation of its function, a breakthrough that led 30 years later, to the development of the revolutionary weight loss drugs that will be discussed tonight. Svetlana is here with us this evening. Svetlana, would you please stand and be recognized? Congratulations on your amazing research. Just this fall, Svetlana also received the Princess of Asturias Award last month in Spain, along with this evening's speaker, Jeff Friedman. These two were among those honored for discoveries that have led to the development of these effective treatments for diabetes and obesity. So all of this brings me to tonight's program and its provocative title, Obesity, Causes and the New Treatments and the End of the Beginning. Unless he changed his title. No, it's still title. Okay. So obesity, commonly defined as elevated body mass index, is one of the most common human health concerns, with now about 40% of American adults overweight or obese. It has not always been this way. It's startling to learn that the average male adult weight in the United States in 1960 was 160 pounds, and today it's over 190 pounds. Women have followed a similar course. And obesity is not benign. It has a number of comorbidities that are attendant to it. Biomedical scientists have labored for decades to dissect how genetics, diet, activity, and other factors contribute to excess body weight and its associated illnesses, both chronic and acute. Our speaker tonight, Jeff Friedman, has made the transformational discovery for the field when in 1994, with heroic work, he discovered the hormone leptin, whose genetic absence leads to massive obesity in mice. As surprising as it was that a single peptide hormone could transform body weight, was his discovery that the hormone was made by fat cells and bound receptors in the brain that regulate appetite and behavior. And moreover, that this gene was not unique to mice, but in fact a nearly identical protein is made by fat cells in humans, as he showed. This revolutionary discovery forced us to change our view of obesity, not simply as a consequence of failure of will, but instead due to a consequence of biological processes that are not simply under our conscious control. I note that Jeff's discovery of leptin at that time was accomplished when discovery of disease-related genes was painstakingly difficult because genome sequences were unavailable and gene mapping, identification, and sequencing was time-consuming, and DNA sequencing at that time was literally 10 million-fold more expensive than it is today. So it was really remarkable that he was able to achieve this discovery. And for the last 30 years, he has remained at the forefront of the field, most recently reporting just last month the discovery of a new type of neuron in the brain that is involved in the rapid induction of satiety. Jeff grew up on Long Island, graduated from Rensselaer Polytechnic Institute, received his MD degree from Albany Medical College, and was a resident at Albany Medical Center before completing a gastroenterology fellowship at Cornell University Medical College. From there, it was a short walk across 68th Street to the lab of Rockefeller professor Mary Jean Creek, who was working on addiction. And this was followed by further work with Jim Darnell, where he received a Rockefeller PhD in molecular biology in 1986. He was sufficiently impressive in his work to be directly appointed assistant professor at the university that same year and began his audacious effort to identify the obesity-causing gene mutations in mice. He's been a distinguished member of the Rockefeller family ever since, as well as a Howard Hughes Medical Institute investigator, and he was named the university's Marilyn M. Simpson Professor in 1999. Jeff's work has received wide recognition, for which he's received many distinguished awards and prizes for his research, including the Albert Lasker Award for Basic Science in 2010. He's also received the Breakthrough Prize and too many other prizes to enumerate all of them. But in addition, he's an elected member of the U.S. National Academy of Sciences and the National Academy of Medicine. In addition to his spectacular talent as a scientist, Jeff is truly a remarkable scholar, a historian of science who has written about many important facets of its history, and a truly wonderful colleague here on campus, always with incisive commentary and creative contributions to important discussions here at the university. I'm delighted that he's here with us this evening to discuss these exciting developments in obesity research and treatment. Please join me in welcoming Jeff to the stage.
I'm not really sure who Rick was talking about there, but welcome everyone. Thank you all for coming. And thanks, Rick, for that really generous introduction. I gave a talk not long ago when they had mistyped the title and it said Obesity Causes and Treatments, the Beginning of the End. And I hope you don't find this talk as apocalyptic as it could have been. Okay, if you were walking on the street and saw two individuals that looked like this and you were asked, or the average person was asked, why the difference, most people would say the individual on the left eats too much. And that would be correct. Nobody gains weight without eating more calories than they burn. But that would ignore the deeper question, I think, which is, why does that individual eat too much? And that simple question, in a sense, has captivated my research interests since the times Rick mentioned. And so I'll talk about that today, first by considering what causes obesity, and then we'll talk about how to treat it, and then what prospects the future might hold. Now, I refer to this as the end of the beginning for a couple of reasons. The first is that in early days of disease research, what causes the disease and the way we treat it are distinct. Think about chemotherapy for a moment. We treated cancers for many, many decades with specific molecules that could arrest or at least slow the cancer, but that had nothing to do with the pathogenesis. But over time, new treatments have been developed that allow us to address immediately the pathogenesis, and these turn out to be much more effective. And I think one of the reasons this is the beginning of the end is that I think in time we'll be able to address pathogenic mechanisms more directly. In addition, from cancer and other disorders, we've learned that these are heterogeneous, that what causes a cancer can be very different in different patients. And I'll try to explore a little bit as we go along the fact that the causes of obesity may be different in different individuals. And ultimately may also require distinct therapies. And then talk about where do we go from here. I have to also begin with a disclaimer. I am about to tell you I'm quite confident more about hormones than you ever wanted to know, and lots of hormones, but hopefully you'll find them pertinent. So if we were going to ask why that individual on the left weighs more than the one on the right, there are three general explanations that are offered in public discourse. The first is that the obese lack a level of willpower that's exercised by lean people, and I find that point of view is more often favored by lean people. Second is that we live in a lifestyle and environment where we have free access to calories and exercise less, and that this is an environmental program. And the last possibility is that obesity is caused by biologic and genetic factors. I'll point out that if I were talking about any other disease, people would immediately gravitate to the idea that there must be a biological basis on which environmental and behavioral factors act. But in this field, for reasons that I think are more sociologic than scientific, people tend to consider one of these explanations to the exclusion of the others. In fact, I once began a talk where I asked people to vote at before and after which of these factors they thought was most important. And I stopped doing that because fewer people voted for biology when I was done than when I began. But I'm going to tell you about what we've learned about some of the biologic and genetic factors that cause obesity. My interest in the area and this notion of what regulates appetite drew my attention to a genetically obese strain of mouse. This is known as the OB mouse or the obese mouse. It was identified in 1951 by George Snell and colleagues, who found that the mouse on the far right has a defect in a single gene that leads it to eat more and weigh 4 times as much as his sibling control immediately to his left that lived in the same cage with the same free access to food and in the same environment. So a single gene can cause obesity and hyperphagia, and the question is, what's the nature of that gene? And as Rick mentioned, in 1994, we found that the gene encodes a hormone. Quick review, what is a hormone? A hormone is a signaling molecule made by one cell, one specialized cell, that circulates in the blood and acts on another cell. It's the way that the cells of the body communicate. Hormones act on specific molecules called receptors. And you can loosely think about this as the way transmitters communicate. There's a transmitter, it emits a signal, in this case an electromagnetic wave. It's received by a receiver, interpreted, and then can influence subsequent events. So hormones are chemical messengers. The leptin hormone is expressed specifically in fat tissue in proportion to its mass. So if you have more fat, you make more leptin. If you have less fat, you make less leptin. Leptin circulates in the blood and acts on centers in the brain in a region known as the hypothalamus to suppress appetite. I'll show you a video in a moment that explains more about how the system operates. But I'll first point out that the OB mouse shown there has a defect in the leptin gene. So it never generates any signal that there's fat stores. The brain interprets this as starvation. The animal continues to overeat, putting on more and more weight, but never generates the signal that would otherwise shut the system off. Now, leptin is a peptide hormone. That means it's a small protein, so it can be made in the laboratory. And working with other scientists at Rockefeller, including in particular Stephen Burley, we made leptin in the lab, and here's what happens if you treat an OB mouse. On the left is a saline-treated animal. On the right is a leptin-treated animal. And you can see dramatic weight loss and a commensurate decrease in food intake. What's the relevance of this to human? Well, this was shown by a colleague in Cambridge, England, Steve O'Reilly, who found a child one day who was of normal weight at birth but began to develop morbid obesity in infancy. The child was massively hyperphagic. There was nothing the parents could do to restrain the child's appetite. At the age of 4, he was already pre-diabetic with high insulin levels and weighed 90 pounds with 57% body fat. Now, this child came from a family with first cousin marriages, and he had a similarly affected 8-year-old cousin who weighed over 200 pounds. Now, when you see an extreme phenotype, as Dr. Leptin has pioneered, an extreme phenotype in an inbred family, it often indicates that there's a genetic cause. And so O'Reilly, knowing about our mouse work, tested this child and found that he too has a mutation in leptin, as did his cousin. And so as a consequence, he began to treat the child with leptin, and here's what happened. This is the child now at 3 and 8. Well, what was obviously driving appetite in this child was not a lack of willpower. It was not a modern environment. It was a lack of the hormone leptin. Now, hormone mutations are poor targets, or hormones are poor targets for mutations. So leptin mutations are rare. But as I'll tell you in a moment, mutations in the pathway on which leptin acts are not rare. But first, I'll say a little more about how leptin acts and what it does. So leptin is made by your fat tissue in proportion to its mass, and at your stable weight, food intake equals energy expenditure. Now imagine you lose weight for one reason or another, such as by dieting. Fat mass will go down, leptin level goes down, and a low leptin level is a stimulus to eat more. And you'll continue to eat more, overeat more, until such time as your fat mass and leptin level return to the starting point. Conversely, if you were to overeat for a period of time or gain weight, and there are many scientific studies supporting this, fat mass and leptin level go up, and in a normal weight individual, that high leptin level will suppress appetite, again returning weight to the starting point. And so what this system does is acts to maintain relative constancy of fat mass. We could talk about this in the Q&A period, but this is actually a system that's under intense evolutionary selection. It's dangerous to either have too little or too much fat, and this system acts to keep the amount of fat each organism carries at an optimal level. I want to emphasize that the leptin system is not the only factor that regulates weight. I'll speak much more about this, but it provides a biological force that resists weight change in other direction, but other factors can also influence it. Now, the child I showed you has a mutation in the leptin gene, and in a sense, this child eats more because his brain thinks he's starving. His brain never receives the signal that there are adequate fat stores and responds accordingly. But most patients, as I mentioned, don't have leptin mutations. In fact, most obese patients have high leptin levels. And you can see that here where percent fat is this not working? I did this once and I pointed in my eye and sort of staggered myself. So I think I'll just use the cursor. What you'll see here is that percent fat increases, leptin level increases. So these people who are the most obese have the highest leptin levels. So most obesity is not caused by leptin deficiency, as is the case in that child. It's caused by high leptin levels. Now, if you have high leptin levels and you're obese, it stands to reason that the leptin you make isn't working well enough. That is an indicator oftentimes of what we refer to as a hormone resistance syndrome. We make the hormone properly, it just doesn't work well enough for reasons we're beginning to understand. Now, if you overexpress a hormone that normally reduces weight, you might wonder what good would it do to give extra hormone? And in most cases, giving extra hormone to a patient with a hormone resistance syndrome doesn't work very well. And that's the case for leptin and obesity. And with this small study, there were larger ones. Giving leptin to obese patients at higher and higher doses, as shown here, may have caused weight loss in some people, but the overall effect size was relatively small, and not everyone responded. So these findings have suggested that obesity is caused by a relative block in leptin action. So imagine leptin is made and its signal is diminished on the brain, less signal would get through, you would eat more, burn less energy, put on more fat, make more leptin, and so by dialing down leptin sensitivity, you would dial your weight up a particular amount. So we, of course, would like to understand the causes of leptin resistance, and that will require understanding the nature of the neural pathways on which it acts to regulate appetite. And an extraordinary amount has been learned about this in recent decades. We now know that appetite is regulated in animals and humans by paired pathways in the brain that either stimulate feeding or inhibiting it. Leptin works by inhibiting the pathways that stimulate appetite and activating the pathways that inhibit appetite. These pathways have generated a lot of attention in recent years because mutations in the pathway that normally inhibits feeding cause human obesity. That includes mutations not just in leptin, but in the leptin receptor and two downstream signaling molecules known as MSH and the MC4 receptor. We now know that more than 10% of early onset morbid obesity is caused by defects in these genes. And moreover, more subtle variations in these same genes cause obesity in the rest of the population. Now, you could think about these genetic defects as being genetic causes of leptin resistance. We block leptin's ability to activate the pathway it normally does, and as a consequence, leptin resistance develops. And more recently, we found that acquired defects that can also cause leptin resistance in mice, and this maps to exactly this same pathway. But because obesity is normally caused by leptin resistance, as I just hopefully communicated, leptin as a therapy is of limited value, at least thus far. And the new obesity treatments do not target directly the leptin pathway. We've also learned in the last few decades that feeding is regulated by other systems. The first, or by at least three different systems. The first I just told you about, it's a homeostatic system whose job it is to maintain constant levels of fat stores. A second pathway is known as a reward pathway. This is the pathway that drives you to eat between meals when you're hungry and to consume foods that we interpret as being pleasurable that generally have high sugar or fat. There's a third pathway known as the satiety pathway, and these are pathways that shut off appetite to complete a meal. So after you eat a certain amount, signals are generated that tell the brain you've had enough to eat, it's time to stop. The new obesity therapies target the satiety system, in particular by mimicking the effects of numerous hormones made by the gastrointestinal tract that normally suppress appetite. So this is a list of some of these hormones, and in general what happens is when you eat a meal, both the nutrient content of the food and the mechanical stretch of the stomach and lower GI tract send signals to the brain. Some of these signals are neural, others are hormonal, and the net effect of them is to shut appetite off and terminate a meal. Now, a number of gut peptides have this property of shutting off a meal acutely. The first of which I'll say much more about is GLP-1, the active form of which was discovered by Svetlana, who Rick mentioned. I'll briefly mention two others, one called GIP, made by the stomach, and another called amylin, made by the pancreas. And these and other of these hormones play a role in shutting off appetite to terminate a meal. Now, the development of these agents, these anti-obesity agents, didn't evolve actually from a search for drugs that might regulate appetite. Rather, they devolved from the wish to develop new treatments for diabetes. And it's because a hypothesis has been extant for more than a century that there's a gut hormone that increases insulin secretion. So when you eat a meal, hormones are made that not only suppress appetite to terminate the meal, but also stimulate insulin so you can metabolize the nutrients you just ingested. And the notion was put forth that if we could figure out which gut hormone it is that might increase insulin secretion, that might be a possible treatment for diabetes. This concept came to be known as the incretin concept, and I'll repeat it here. You eat a meal, nutrient in the intestine and/or stretch stimulate the secretion of intestinal hormones, which were generically known as incretins. Incretins both shut off the appetite and end the meal, as well as stimulating insulin secretion to reduce blood glucose. So you eat, your glucose would normally go up, and this provides a mechanism for maintaining blood glucose in a healthy range. And it was not lost on anyone's attention that incretin, or the incretin, if found, could provide a new treatment for diabetes. And decades of research ensued trying to find this gut hormone that might regulate insulin secretion. A quick review of insulin. Insulin acts to maintain stable levels of blood glucose. Normally, glucose is sensed by pancreatic beta cells. It's a hormone. Glucose is sensed by beta cells. The hormone is made. It acts on a variety of other tissues to lower blood glucose. Defects in this pathway cause diabetes. In type 1 diabetes, you fail to make insulin, and as a result, blood glucose goes up. But in type 2 diabetes, which is present in 90% of diabetic patients, there's a defect in insulin signaling. It's another hormone resistance syndrome associated with high insulin levels. Insulin is a fairly good therapy for type 1 diabetes, though imperfect, but it's a less effective therapy for type 2 diabetes for the same reasons I mentioned for leptin. And for that reason, new therapies were needed, and this was the main driver to try to identify incretin. Well, the discovery of incretin devolved from yet another hormone I'm afraid I have to tell you about, named glucagon. Glucagon is a hormone also made by the pancreas in response to low blood glucose, and it acts to raise blood glucose and counteract hypoglycemia. Well, glucagon has a number of interesting features in its own right, and it was studied and found in the 1980s to be produced by what we call a precursor protein. It's a longer molecule that's chopped up into pieces that have different activities. And what was found in 1983 in hamsters by Graham Bell and colleagues is that this glucagon precursor molecule is cleaved to produce multiple hormones, not just glucagon, and it was later shown that it was by Svetlana that it was differentially cleaved in gut and pancreas. In gut, it was cleaved to form not glucagon, but a molecule called GLP-1 to 37. In pancreas, it was cleaved to form glucagon. Well, when people saw that there was a gut hormone, GLP-1 to 37, produced in response, as it was shown, to nutrient, they said, well, maybe this is the missing incretin. There was, however, a problem. GLP-1 to 37 had no effect on insulin secretion. And many at that point abandoned it, the study of this molecule, except for Svetlana. And Svetlana had reason based on evidence that she'd be much better able to share than me, that GLP-1 to 37 was processed further and that it wasn't the active molecule. And what she proceeded to first propose and then show is that this GLP-1 to 37 is further cleaved to a shorter form, 7 to 37, only in the intestine. Well, now there was an opportunity to test GLP-7 to 37 and see if it stimulated insulin secretion. It could be used to treat diabetes. And that's exactly what she and colleagues, David Nathan and Joel Havner, did. And here's what happens if you infuse GLP-1, giving it as an intravenous strip, into diabetic patients. Shown here in blue is insulin goes up, glucose goes down. This is an incretin. It has other features of incretin I won't go into. So this drug, or this hormone, they showed has very potent anti-diabetic effects. It's the missing incretin. There's a problem, however. This molecule has a normal half-life of 3 to 5 minutes. That's why you have to infuse it. If you just give a single injection, it'll be gone before it has any effect. And so in the pharmaceutical industry, intense efforts that a number of companies developed to make a form of GLP-1 that would last more than 3 to 5 minutes in blood, in fact, last long enough to have a sustained effect and obviate the need to infuse it over a period of time. Well, the most effective of these agents for the treatment of diabetes was developed by Novo Nordisk first, a molecule that you may have heard of as Victoza. They did something rather ingenious at Novo Nordisk. They stabilized GLP-1, and from now on, when I say GLP-1, I mean GLP-1 7 to 37, the truncated form that Svetlana discovered. They modified it by adding a fatty acid. It's sort of shown here as a cartoon. When you add a fatty acid, it binds to albumin in blood, and this stabilizes it. It protects it from excretion by the kidney and also protected it from degradation by enzymes. And now the half-life increased to 12 hours. And here's the effect in diabetic patients. It was actually quite remarkable. The drug increases insulin, lowers glucose, lowers hemoglobin A1C, an indicator of diabetic status. And based on this, it was approved as an anti-diabetic agent in 2010 and has had a tremendous impact to improve the health of diabetic patients worldwide. Now, the drug had to still be administered once a day, and patient compliance is often a major problem. And a once-a-day injectable is not a particularly desirable profile for a drug that patients are going to take for a sustained period. So as is often the case, Novo Nordisk now set out to make an even more stable form of GLP-1, one that perhaps could be administered once a week. And they succeeded in doing so with some even more novel chemistry, the nature of which I won't tell you about. And this drug is called generically semaglutide, but goes by two names you may also have heard of, Ozempic and Wegovy. And this is a GLP-1 7 to 37 that's stabilized with a modified fatty acid with other amino acid changes, and it has a half-life of 7 days. Now, when this drug was developed at Novo Nordisk, it was with the expectation that it would just be used for diabetes. And I was told by the people who led this program that they were as surprised as anyone when they saw the following, which is when they gave this extended half-life GLP-1, they saw dramatic weight loss. Average weight loss of 12%. Now, this drug acts to stimulate the satiety system. The satiety system is what makes you feel full over the course of a meal, and then you overeat to an even greater extent, might even make you feel nauseous. And the drug acts largely by inducing feelings of fullness, often advancing to nausea. But nonetheless, people seem to get used to it, and the weight loss result was profound. I want to digress for a moment and emphasize this point that it doesn't address the cause of the problem, because these effects are pharmacologic. Physiologic increases in GLP-1 levels don't alter food intake or weight, and mutations of the GLP-1 receptor or GLP itself don't affect weight. This is what's known as a pharmacologic effect. It's not a physiologic function of the hormone. But nonetheless, it has had and will continue to have, I believe, a dramatic impact on human health. It's a blockbuster treatment for obesity and diabetes. It reduces the risk of heart disease, and very importantly, it does so not just in diabetic patients, but even in obese patients without diabetes. There are recent reports that it can preserve kidney functions in patients with impaired renal function, and there's also even newer data that it can even have benefits in Parkinson's disease or alcohol or other types of addiction. So we're going to learn more about the potential health benefits of these drugs, I think, in the coming years, but there's a tremendous amount of excitement about their potential. Some of you may have seen an Economist cover recently that said, I think, the everything drug. But the story doesn't end there, because there are new agents that have been since developed that are actually even more effective, at least for reducing weight. And what these are hybrid hormones that act simultaneously at multiple receptors for these different gut hormones. The one I think you may have heard about is what's known as Mounjaro or Zepbound, developed by Lilly. And this is a modified hormone that can mimic the effects not just of GLP-1, but another of the hormones I mentioned, GIP. And this combined, what's referred to as dual agonists, this concept was pioneered by other scientists named Matthias Chup and Richard DeMarchi. And what they showed is that Zepbound is even more effective than Wegovy. It causes 18% placebo-corrected weight loss and even seems to have less nausea associated with it. But despite the efficacy of this and other hybrid hormones that are being developed, the story is unlikely to end here, and there are still opportunities, I think, for further innovation. Well, for one thing, while these drugs have a tremendous overall efficacy to reduce weight, they don't treat everybody. Some people cannot tolerate the side effects. Other people suspend the drugs for other reasons. And what this slide shows is that while more than 50% of patients show greater than 20% weight loss, 30 to 50% of patients who are treated don't meet their weight reduction targets. And that's shown on these data, which are derived from the paper reporting these results, and simply shows that the number of people who don't meet their target weight can be as high as 30 to 50%, depending on the dose. There are other limitations of these drugs, and I don't mean to say they're highly effective and a major advance, but every drug has issues associated with it that need to be managed. There are tolerability issues due to nausea and other GI side effects that can be more severe in some patients. There's also a temporary paralysis of the stomach, and some of you may have heard that surgeons suggest that patients stop taking these drugs before surgery for fear of gastric motility problems and vomiting. The drugs cause a loss of muscle mass, the significance of which is not known, but is at least of theoretical concern in older patients who lose muscle mass anyway. There are anecdotal reports of suicide, eye issues, and other complications, but I'll also point out these drugs have been in patients for diabetes for more than 2 decades now, and there don't appear to be any major side effects or safety flags. In addition, you need to be on these drugs permanently. If you withdraw the treatment, weight rebounds to the starting point. So I think there's an opportunity to develop new therapies, either for weight loss or weight maintenance. And I'll close by simply throwing out some ideas about where we go from here, beyond making more and more hybrid peptides. And I think there'll be opportunities in time to also target the homeostatic system on which leptin acts. I already showed you evidence that leptin can be highly effective in a setting of leptin deficiency, and there are many other examples of this I won't talk about. Diseases known as lipodystrophy, hypothalamic amenorrhea. But one might also wonder if there are other patients who have low leptin levels in obesity who don't have mutations. And if you look back at the slide I showed you before, you can see evidence for that. So if you look here, here's weight related to leptin level. Boxed are patients who are quite obese but have relatively normal leptin levels. And so the question was extant, might these patients respond to leptin? If high leptin levels indicate leptin resistance, might a lower leptin level indicate leptin sensitivity? And Amgen, who performed this study, actually tested this directly. And what they did was they pre-selected obese patients with low leptin levels in this range. And here's the result. If you treat now this subset of obese patients, it turns out to be about 1/10 of the population, you start to see a little bit of a lag, but over the course of 32 weeks, you see highly significant weight loss of about 12% with no evidence yet for a plateau. And in contrast to these other agents, leptin appears to spare lean body mass and does not have any of the side effects associated with these drugs. So they might be better tolerated if we could target the correct population. So with respect to the notion that obesity might be heterogeneous, I think this and other evidence suggests that indeed obesity is. Much the same as diabetes can be caused by absent insulin or insulin resistance with high levels, I think the evidence is that obesity can be caused by low levels resulting from mutations or other things that such as transcription defects that lead to absent or low levels. And we might think about this as type 1 obesity, for which leptin in time might prove to be an effective therapy. Type 2 obesity is similar to type 2 diabetes. There are high levels. It's a hormone resistance syndrome. Other therapies are needed. And for this, the GLP-1 agonists have proven very effective as well as these other dual agonists. But I'll close by telling you that it may be possible in time to develop cell-specific neuromodulators that modify the activity of the neural circuit I showed you. And I'll close by showing one last data slide that if you combine GLP agonists or these dual agonists with leptin, you see a synergistic effect even in human. Here's the animal data for leptin plus one of the dual agonists. Here's leptin alone. It doesn't work very well in leptin-resistant animals. Here's the GLP agonist, dual agonist, weight loss. You add leptin on top of it, you see a synergistic effect. And the same has also been shown in a human study combining leptin with another peptide that I mentioned, amylin. Leptin and amylin alone had showed mild weight loss, but the combination showed a highly synergistic effect. So I think this is a time of great promise and excitement. I think there's be an opportunity to now subsegment obese patients and tailor therapies to them, and perhaps combine different therapies to get synergistic effects. So I'll stop here and tell you the key points I hope to have communicated tonight. The first is that defects in leptin signaling are a cause of obesity, and probably the evidence suggests the main cause of obesity. But three systems control food intake, not just the homeostatic system of which leptin is a part, but reward pathways and satiety pathways. The new therapies reduce weight by inducing satiety via pharmacologic effect. But I think it leaves open the possibility that there may in the future be opportunities to target the homeostatic system and the pathogenic mechanisms that lead to obesity. So I'll stop here, and I'd be happy to take any questions you might have. What?
So
I think I spoke longer than I was supposed to. Sorry about that.
Everything's great. That was a wonderful talk. We have roving microphones, so if you have a question, please raise your hand. Keep it up long enough for somebody to recognize where you are, and a microphone will get to you. And we have a question there, another question down here. Other people, raise your hands if you have a question. Okay. Go ahead.
Thank you very much for that fascinating talk. And I wanted to ask two questions, please. I know there have been a mass of studies about the application of these drugs in heart, kidney, and other such chronic diseases. How credible do you think they are as in terms of reducing alcohol addiction, nicotine consumption, firstly? And secondly, could you talk about the anti-inflammatory properties of these products? And to what extent are the different GLP-1s homogeneous in that respect? And how significant do you think anti-inflammation is in terms of heart?
So these are very sophisticated questions. So with respect to the first, there are clear connections. So these hormones, the SI system is centered in the brainstem, but they communicate with reward centers, which are largely localized in dopamine pathways in the midbrain. So there's direct crosstalk between them. I don't think it's implausible that there could be effects on reward pathways that could account for these somewhat descriptive studies showing reduced addictive behaviors. The best evidence that I know of is for alcohol, and I'm not entirely certain to what extent that's been dissociated from the caloric intake and the reward value of alcohol. Alcohol has a lot of calories in it. The links to Parkinson, one can hand wave and say it's also dopamine pathway. I think that link is a little more tenuous. I think the reward addiction pathways is more plausible, in part because some patients who take these drugs report what's known as anhedonia, general lack of feelings of pleasure. In terms of the anti-inflammatory effect, it's a credible and attractive hypothesis to explain all the different apparently beneficial things these things do. There's very limited evidence, however, to support that. There's one recent paper in mice that says there might be an anti-inflammatory effect, but I think a lot more work will need to be done to establish that. Of course, if there is an anti-inflammatory effect, it could account for some of the reduced heart attack risk, but losing weight also, it would seem to reduces risk. So I think the role of anti-inflammatory effects for this or other molecules in the class really is going to require a lot more research.
A question here. Another question down here.
Yeah, thank you. I was wondering whether the newer drug, whose name I forgot already, that seemed more effective than Ozempic at all, does it hint at any of the other side benefits, say to heart and kidney?
We don't know yet. So Wegovy was introduced earlier, so there's more data on outcomes. I'm sure the outcome trials are underway for this drug. Frankly, I'd be surprised if they didn't have a similar health benefit, but we can't say with certainty yet.
Question here and a question here.
Thank you. It was incredibly interesting, a little higher level science than I've been exposed to in decades. But my real question is a few comments about demographics. So what makes first of all, are there studies about the loss of leptin in the general population? I mean, you're looking at obese people and you're finding this. Do we know in the general population, are there thin people with these same inhibitors? And then the second question is, so why is it different now than it was 40 years ago?
Well,
and the third part of that, one last thing is,
are
there different populations, demographics? Do we know anything about that too?
Yeah, so I'll start with the last question. So different populations have different propensities to obesity. I hypothesize based on some evidence that it has to do with the evolutionary environment in which that population developed. Being too heavy is a potential risk. Being too thin is a potential risk if you're in an area where famines are prevalent. But being too heavy is a risk if you're in an area where predators are a problem, historically. And this is true in animal species too, and you can draw your own conclusions about why that might be. But particular populations, I think, regulate their adiposity at a particular level to balance the prevailing risk in that population. So, for example, the most obesity-prone populations on Earth are Pacific Islanders, where famine was always a major problem because of typhoons and predators were not around. So that explains the demographic difference. As far as secular trends, notwithstanding Rick's comments, if you normalize for age and sex, the changes over this period have been less dramatic than you would think. Now, you mentioned men only, but you also need to normalize for age. A population gets older. People tend to get heavier as they get older for reasons we don't understand. And what's happened, I think, in the population is this. As average weight increases, we define obesity as a BMI greater than 30. And it's a bell-shaped curve, sort of. And if the curve shifts and the threshold doesn't change, then you can get a disproportionate number of people who exceed the threshold with a relatively smaller weight change. And the evidence that I was provided and tabulated with an NIH epidemiologist says that actually accounts for the massive increase in obesity that we hear reported on. Because if you normalize for age and sex between 1970 and 1991, the average weight change associated over that time was 7 to 10 pounds. And I think that number is closer to what I think the environmental factor is. I think the differences in a cross-section of the population at any one time is genetic. So I think, and the environmental factors to a large extent may simply have to do with the fact that everyone now has, or more people have free access to calories, whereas previously some people had an inability to consume as many calories as they wanted.
So a question there, and then here, and then here.
I wanted to ask you about the onset of the obesity. If a child were of normal weight for most of his or her life, and then in the 30s began to get very heavy, would that suggest that these hormonal dysfunctions began, or does that suggest that it's more an emotional variant? Because in your first slides, you'd pointed out that the infant child already showed that there was a situation. So could you comment on that? And secondly, could you comment on at what age would it be inappropriate, like a child or a young adult, and that other sources of therapy or nutrition be undertaken?
Okay, so again, I'll start with the second question first. Leptin mutations are very rare, so I don't think there's any urgent need to measure leptin levels in children to make sure if they have leptin, whether they might have a leptin mutation. The one exception to that would be if you have a very obese adolescent who didn't enter puberty at the normal age. That's seen in these patients, and that might be a clue that you might want to think about measuring leptin level. The first question you asked is one that I thought about a lot, and I wish I had a better answer. That is, why is it that there are some people who are at a stable weight, and then all of a sudden their weight takes off? And it happens. I have at least one theoretical explanation that might very well be wrong, but would be testable. I've just not been able to see my way clear or figure out a way to test it, which is this. We're learning increasingly that there are certain neurologic disorders that can be caused by selective loss of a neural population. And the best example of this is Parkinson's disease, but there are others. Some of you may know that narcolepsy, a sleep disorder, has recently been discovered to be a result of a selective loss of a very specific neural population in your hypothalamus. So one possibility for this, which is purely speculative, is maybe you lose neurons in this POMC pathway. So I find that satisfying only insofar as at least could provide a potential explanation for this. And it is testable if one could access necropsy samples and look for it, as was done for narcolepsy. There's also evidence, and I only alluded to this with the reference to an acquired acquired leptin resistance, it's beginning to become clear that as leptin levels go up, in some patients, that downregulates the leptin response. So it could be that in some patients, they tend to gain weight as they get older, but then they pass some critical threshold where now that higher leptin level has downregulated the response to such an extent that they now reset it at the higher weight. And if in that manner, they could keep ratcheting up. And I don't want to prattle on too long about this, but afterwards I can tell you why I think that's plausible in the reception.
I promise this will be a one-part question. And it's simple. Are the added benefits to GLP-1 generally, the general class of GLP-1 drugs, enough to, over time, warrant the general population to take them as a preventative measure for Parkinson's and the other things that you said that were, possibly benefits that keep being discovered?
Well, I think the clearest example of that really is this protection from heart attack in patients who are not diabetic. There are a lot of obese patients, and I think it's an open question is whether we should be treating obese patients who don't have comorbidities such as diabetes, hypertension, known heart disease, whether we should be treating those patients prophylactically. I think that's a case where the evidence is such, and Rick is a better clinician than me, so he may want to weigh in, but that's a case where it's a serious question. As for whether it has preventive value for Parkinson's or Alzheimer's, all these other things, I just don't, the data aren't there yet. But I think the general question is very pertinent based on what we already know.
But only in people that are obese?
Well, that's where the evidence is now, so it raises the question, should every obese patient be on this? I probably would, that's not even a question I'm equipped to really answer. I think it's a legitimate question.
Question right there.
Sort of two-part.
It's like what scientists do. When you're giving a presentation and they say 4 slides, you show 4 slides with like 6 animations each.
So my 39-year-old daughter, who was too busy seeing a show tonight and didn't come, has said, well, we don't know the long-term effects of these drugs, and so why would I do it? That's one. And two, once you are successful and do stabilize your weight at a low level, are you taking a $1,000 a month drug for the next 50 years when you're not under insurance?
Well, I would say that might have a lot to do with how much money you have, but setting aside the obvious, I think that's the latter is a case where we'd really like to find new therapies, and it may be that the ways we maintain weight loss will in time be different than the ways we induce it. You're correct. We don't know what the long-term safety of these drugs are. The drugs have been in people for decades, but at lower levels than they currently are. And there are receptors for these things everywhere, in many, many places. And so, so far, all the effects seem to be positive, but we'll find out. I tend in general to be less concerned about safety of drugs that are hormone mimetics than, let's say, small molecules, but that's not a guarantee.
Questions?
Good evening. Thank you. Do you have a minute to speak about your BDNF studies, or shall I catch you at the break?
Well, I think I can answer it as follows. So we all think about feeding as like this incredibly complex thing. I made it seem incredibly complex, and it is in a way when we talk about 3 different systems and many, many factors that regulate it, the behavior. But every vertebrate regulates their feeding perfectly well, and fish and reptiles, for example, don't have a cortex at all, and they regulate their feeding with great fidelity. largely through the hypothalamus. And so there was a Nobel Prize-winning scientist in 1906 named Sherrington. And what he postulated was that behaviors, even complex behaviors, are regulated by very simple circuits that resemble a reflex. And this is then the substrate for all the other factors, including willpower, that regulate behaviors. And the example he gave for this is a cough reflex. When you're in an audience, a cough reflex is like a knee jerk or any other reflex. It's a 3-neuron circuit, but we can consciously control our cough reflex to an extent. So if you're in a crowded theater, you may be able to suppress your cough. So he proposed a model that complex behaviors are regulated by simple reflex-like circuits that evolved in lower animals that are conserved, but then are the substrates for more and more complex processing as the brain develops further. And we were interested in whether this concept applied for feeding, so we wanted to map what the simplest circuit we could find is that regulates appetite. And we found, as he would have predicted, a simple 3-neuron circuit that includes a molecule named BDNF that can regulate appetite. So it's an opportunity now to explore how all the different factors that regulate feeding change the behavior, perhaps by modulating the activity of simple circuits like the one we described.
All the microphones getting to this gentleman, I'll ask a question. So you emphasized that GLP-1 has a half-life of 3 to 5 minutes, but the effects of the drugs are pharmacologic at vastly higher doses. What do you think the receptors in the brain are there for when they never see a dosage of GLP-1 that ought to be adequate to achieve much effect?
So it's a two-part answer to your question. There are, it's my turn. So these drugs will acutely reduce appetite, and there's a part of the brain that doesn't have a blood-brain barrier, and there are receptors for this there. So I think the short-term effects of these agents are on those accessible receptors, but that doesn't explain the full effect of the drugs. It turns out there are populations of neurons in the brain that also make GLP-1, and those neurons suppress appetite by sending outputs lots and lots of places. And so there's GLP-1 receptor all over the brain that's targeted by GLP-1 in brain that has nothing to do with these gut signals at all. And I think part of the mechanism of action of these drugs is by acting at those sites which are not normally targeted by the native hormone. But something about the high levels that these drugs achieve and or the lipidation allow it to get access to deeper structures that they wouldn't normally get to.
Amazing. Thanks.
The question is, if some persons, regardless of ages, have the capacity to regulate their eating habits themselves and use exercise and have success in this area, does that suggest that it is possible that some of the combination of exercise and diet is capable of creating the chemical neurological reactions that you talk about? Is that feasible, or is it luck, or is it just happens to be occasionally?
So actually, I'm particularly glad the person a moment ago asked about BDNF, because I think it's pertinent to the answer to your question. So first of all, exercise by itself is not a very effective way to lose weight. Diet and exercise is what's been prescribed since the time of Hippocrates, and it's never been shown to be efficacious in anything other than a small percentage of the population, on the order of 5 to 10%. Now, that's not insignificant. 5 to 10% of people can use cognitive effects to suppress appetite. Now, in this case, what you have is a tension, as is the case for many other things, including addiction, between a biological drive and a conscious wish. And they're often in tension. And my experience, or at least my opinion, is that invariably the basic drive wins, but not in everyone. And what I think it's telling us is that we have these basic circuits that drive appetite, but we have a cortical representation that might say, I really want to lose weight. I am not going to eat. I'm going to resist that drive to eat. Now, it may very well, now willpower is not metaphysical. So maybe the connections between your cortex and the lower centers that regulate the basic drive are different in different people, and that some people can use willpower and some cannot. And so I think this notion of a simple circuit that's modulated from above is attractive to me because it may give us an opportunity in time to understand how more complex representations like the conscious desire to eat less why it might work in some people rather than others. There's no evidence that diet and exercise work through these factors, however.
Last question up here.
I was wondering, in a genetic obesity syndrome like Prader-Willi syndrome, would GLP-1 or amylin and leptin be helpful in these children?
I'm trying to remember. So Prader-Willi appears to be the result of some dysfunction in the hypothalamic region, the pathways I talked about that receive these signals. And I think the answer to the question of whether GLP reduces weight in these patients is known, but I can't. What I'd like to say is I don't think they work as well in these patients, but I don't know that for a fact, and so I'll look it up later.
Well, we could go on for another hour, but I suspect we should draw this to a close. Jeff, thanks for a wonderful talk and discussion. And thank you all.